This is the glucose and energy clip. This slide is looking at the glucose sources for muscle contraction. Remember, in the last couple of clips we looked at glucose and how it can be used to produce ATP for muscle contraction. Now we're looking at the sources of that glucose for the muscle. The glucose can come from blood glucose or it can come from muscle glycogen. If the glucose is sourced from the blood, it could be from exogenous sources, and what that means is outside of the body. When we drink a liquid that contains glucose, that glucose is absorbed from the digestive tract and then into the blood. The glucose in the blood can then be taken up by the muscle and used in glycolysis. Blood glucose can also come from liver glycogen. So the liver has a number of functions, and one of its functions is to store glucose as glycogen. This diagram here is showing you a glycogen molecule with every one of these individual green structures shown being a glucose molecule. And a glycogen molecule can contain hundreds to thousands of glucose molecules. So this glycogen can be broken down in the liver into individual glucose molecules, and then that glucose released into the blood and then taken up by the contracting muscle. Gluconeogenesis is another mechanism where the body can supply blood glucose, and this term refers to the making of new glucose. It does occur in the liver, and I'll talk about that more in the following slide. So basically the liver can take other materials, produce glucose, and then release that glucose into the blood. So it doesn't matter what the original source of blood glucose is, the important point is that it can be taken up by the muscle and used to produce ATP. Glycogen is also stored in skeletal muscle, and when the skeletal muscle starts contracting, that glycogen can be broken down and the individual glucose molecules used for ATP production. So two terms you need to be familiar with: glycogenolysis and glycogenesis. They do sound very similar, so it's important that you understand what the differences are. Glycogenolysis is the breakdown of glycogen. So given that the glycogen is stored in the liver and the muscle, this process is the breaking down of glycogen into individual glucose molecules. The liver can release individual glucose molecules into the blood. However, when skeletal muscle breaks down glycogen, that glucose is retained within the muscle. There's an enzyme that is present in the liver that's not present in the muscle that breaks the glucose apart from a phosphate. This breaking of the glucose apart from the phosphate releases free glucose, and as I said, the liver can release the glucose. The muscle doesn't contain the enzyme that breaks the phosphate apart from the glucose, and therefore it remains trapped in the muscle. Glycogenesis is the opposite, and that's the making of glycogen. So that's when individual glucose molecules are bound together to form glycogen. An important point to note is that glycogen that's stored in the liver and the muscle is depleted within a few hours of prolonged exercise. So in terms of comparing it to fat stores, generally speaking, if you look at typical levels of fat stored in the body versus glycogen, there's a lot more energy stored in the fat stores when compared to glycogen. And you might have heard of carbohydrate loading, and that's when athletes who are involved in prolonged exercise, for example marathons, triathlons as another example, they do what's called carbohydrate loading, and that's where before the race, using the diet, they try and load as much glycogen as possible into the liver and the muscle. So there's more energy stored as glycogen for those long races. It's also common for athletes to consume sports drinks during a race, and those sports drinks contain glucose. So that's to keep those blood glucose levels up to supply the muscle with a fuel source for the production of ATP. Now on to gluconeogenesis, which I mentioned a couple of slides ago, and remember that that's the making of new glucose, and it's the making of new glucose from non-carbohydrate molecules. The molecules listed here are raw materials for the making of new glucose. So amino acids, which remember are the building blocks of proteins, lactic acid, which remember is the end product of anaerobic glycolysis. For a long time it was seen as an end product that was a waste, but as we'll see in this slide and some other slides in the topics to come, that there is energy stored in the lactic acid which we can access and use again. Also glycerol can be used to make new glucose. Glycerol is part of the triglyceride molecule, so in other words, part of a fat molecule. So remember, glycolysis was taking glucose and breaking it down to lactic acid in anaerobic glycolysis, or as far as pyruvic acid for aerobic metabolism. What these molecules do is they're taken through the reverse of glycolysis, or the reverse of part of glycolysis, where they're then formed back into glucose. So for amino acids, they are converted into pyruvic acid and then taken through the reverse of glycolysis up to glucose. Lactic acid, remember, is going one step further in the glycolytic pathway, where pyruvic acid is converted into lactic acid. So the lactic acid is taken through the reverse of glycolysis, where you get the production of glucose. Glycerol can be converted into what we call an intermediate in glycolysis. What that refers to is one of the substrates in glycolysis, and I'll show you this on a diagram in some following slides. And it's taken through then the partial reverse of glycolysis. So it's not taken through the whole pathway, but approximately half of the pathway. So just to sum up, gluconeogenesis is the making of new glucose from non-carbohydrate sources, and amino acids, glycerol, and lactic acid are all materials used for the making of new glucose. Once this glucose is formed in the liver, then it can be released into the blood to be used again as an energy source. And tied to this is a concept referred to as the Cori cycle, and that's the two-way traffic of lactic acid between the muscle and the liver. So remember that during intense exercise, when the muscle's contracting, relying on anaerobic glycolysis with lactic acid being the end product. So remember, I took you through examples, say the 400-metre race or the 800-metre race. Because it's relying heavily on anaerobic glycolysis, there'll be a large amount of lactic acid produced. This lactic acid can then enter the circulation and be taken up by the liver. That lactic acid can then be taken through the reverse of glycolysis to produce glucose. It can then re-enter the blood as glucose, be taken up by the muscle again, and used to produce ATP for muscle contraction. So it's recommended that you attempt the quiz on LMS now, called Glucose and Energy, and when you're happy with your understanding of the content in this clip, you can move on to lipids, protein and energy.